Hello everyone, I'm Inayat Batish. I'm from Food Science and Technology Department from Virginia Tech, and today I'll be presenting a part of an offshoot of my research, basically. The six-word summary for this is basically the superbugs in the bay and how it affects us and the mankind. So before getting into that, we must try to understand how it affects us. The most important thing that affects us is food. And one of the most important source of food is basically seafood. Why is that? It's because it is a highly consumed commodity. I'm sorry, the slides are kind of wonky. It is a highly consumed commodity. As you can see from this graph that we are eating more fish than we are reproducing each year, especially from 1950 to 2015. It has a really high global production that ranges up to millions yearly. It is the most traded commodity all over the world, which although is projected to be approximately 200 million in 2027. And even in 2019, it is approximately $160 million. It accounts for a lot of economy. And a lot of people consume seafood all over the world. It has a huge per capita consumption, as we can see from all these graphs. So the crux of all of this slide is that seafood is really important for the sustenance and for humankind, for food as a food source. Now, talking about the sea and what other resources we take from sea and what are the other seafoods, as we can see from in the United States, billions and millions and billions of economy and production of seafood is done. The major players for seafood is mussels, shrimp, salmon, and clam, but one of the most important seafood is oyster, which accounts for a whopping $106 million of economy and is produced 36 million pounds. Now, why are oysters important as for seafood and in general? So oysters are natural water filters. They concentrate a lot of dirt, a lot of microorganisms, and they clean the water, which keeps the ecology going. Because of this, they have earned the title of habitat creators, also known as keystone species. Now, without oysters, the ecosystem ceases to exist because it has a lot of predators and it cleans the water. Third, it is really important to the Chesapeake economy, especially for the Chesapeake Bay, because it raises millions in the economy and has grown a whopping 24% just in 2012. Now, the problem arises with the microbiota of this bay. Now, microbiota is composed of a consortium of microorganisms. These are viruses, bacteria, algae, and fungi. Now, the inherent nature of oyster is to concentrate them, to filter them. And because it concentrates all of this microbiota, it can pose a significant threat, especially for disease-causing microbes. Now, one of the most pathologically important disease-causing microbes is the bacteria, especially the Vibrio species. Out of this Vibrio species, 2 are the most important, which cause a lot of diseases throughout the world. That is Vibrio parahemolyticus and Vibrio vulnificus. Now, why are they important and why do they cause a lot? How much disease do they cause and how do they affect the economy? One of the most important points is that Vibrio parahemolyticus and vulnificus are cause of one of the most significant and most common cause of outbreaks globally, not just in the States. They put millions of dollars of burden on the government so that they can recuperate and they can provide aid to the people. It is one of the most common cause of shellfish diseases. So anybody who eats oysters or consumes it has to face Vibrio species or Vibrio species diseases. And it was able to increase 57% foodborne illnesses just by itself. So tackling Vibrio species becomes really, really important. But as we know that it is basically the natural part of microbiota. So why is it so important? Why can't we just process the food and why it is becoming a problem? It becomes a problem when it turns into superbugs or basically antibiotic resistance. Now we all know the basic, like when microbe enters our body, we take antibiotics. and the microbe dies. But to escape his fate, microbes have come up with a clever strategy in which they can change their DNA and render the antibiotic inactive, and they're able to live. And once they're able to live, it hampers the disease curing capability of hospitals. It causes multiple deaths, and it also hampers the economy because people will not consume the food that is afflicted with antibiotic resistant bacteria. Just in USA, 2.8 million infections are caused by these superbugs or antibiotic resistant bacteria. 35,000 people die, and it puts a pressure economy-wise on the government of $1 billion, specifically 4.6 billion yearly. Now, why do this antibiotic resistance occurs? The main reason for antibiotic resistance is just basically the overuse of antibiotics in a lot of areas. Once the concentration of antibiotics increases in their environment, which is usually due to us because we overuse them, it is forced to mutate and become antibiotic resistant bacteria so that it can live. Basically, it is a living, or it's basically a strategy for it to elongate its life. So increase in antibiotics causes this effect. Now, this antibiotic resistance can be transferred from either bacteria to bacteria due to reproduction, or it could be due to an antibiotic-rich environment, which this bacteria faces. Now, antibiotic resistance can spread through multitude of ways, as we can see through this really good infographic, that if antibiotics are concentrated in animals, which we feed them so that they don't get diseases, their gut microbiota starts developing antibiotic-resistant bacteria. And once they become our food, or basically give any byproduct, it enters our system, and the drug-resistant bacteria or superbugs are created. On the other hand, the antibiotic abuse, which is done by hospitals, that is unnecessarily prescribing antibiotics when it could have been handled in a better way, it enters the patient, we transmit it, and then it again ends up in our system, and we again are filled, and we again enter a system that is filled with antibiotic-resistant bacteria. For the oysters in the Chesapeake Bay, the main reason for antibiotic-resistant bacteria is basically the overuse of antibiotics that the industry uses. And because they overuse it, the antibiotics are concentrated in the water and in the wastewater. And once they run off and they are diverted, the antibiotic-rich conditions are transferred all over, and we get multitude of antibiotic-resistant bacteria. So this is where my research focus comes in. What I try to do with this small offshoot of my research, and which is currently going on, is to test the prevalence of these antibiotic resistance bacteria for Vibrio species. Now, the first part that we did is we isolated them from these oysters. We collected these oysters from James River, and we isolated these specific Vibrio species. Next, we tried to characterize them. I tried to characterize two most main imparting Vibrio species, that is Vibrio parahemolyticus, L. magnificus, as we discussed before, which is responsible for majority of outbreaks and diseases throughout the world globally. And then we tried to test its antibiotic susceptibility, whether it is antibiotic resistant or not. Now, over 100 of these Vibrio colonies were basically isolated, but we identified and worked with over 40 different Vibrio colonies. To be precise, there were 47. So just as basics, not without going into a lot of technical details, what we did was we took the oyster meat, we homogenized it, and we grew it on a media. For a microbe, a media is like food. So without the media, the microbe cannot grow outside of its host. So this specific media that we used is called the TCBS agar. So the TCBS agar basically has food. It has certain chemicals that will inhibit the growth of unnecessary microbes that will compete with Vibrio for food. And it has a really high salt concentration, which mimics its natural habitat, which is the sea, in which there is a really high salt concentration. This aids and helps the Vibrio to grow. So this is basically how we isolated these Vibrio colonies. These are the pictures from lab. And as you can see in here, there are like multitudes, so many colonies of bacteria. We tried to pick up those colonies which were the most isolated and which were the easiest to pick and worked with them. As you can see, this one is like really clouded and this one is too. Some of the samples didn't show as much growth, but all in all the oysters had pretty good growth of Vibrio, which tells us that Vibrio is very prominent in the Chesapeake Bay, especially in the James River area. Now, the next thing that we did is we tried to characterize them. And what conclusion we came to is that most of them were actually Vibrio vulnificus. Vibrio parahemolyticus was not as common, at least in the James River area. So to test this, what we did was we isolated some colonies from the previous step, and we grew them on a specific nutrient agar, specific food that is called the hardy chrome agar. So hardy chrome agar is basically a chromogenic agar. What it does is that once bacteria grows into it, based on how biochemically it degrades the food, it will change its color. So Vibrio parahemolyticus will be assumed a teal color or a turquoise color, and Vibrio vulnificus assumed a purple color. So that is how we differentiated. And overall, we were able to find that almost 77% of the isolates that we had were Vibrio vulnificus, and a small percentage was Vibrio parahemolyticus, that is 21%. And we found another microbe there that was called Vibrio alginolyticus, but it is not as important disease-causing microbe, so we considered it in our study, but we didn't do further studies with it. So these are the pictures from the lab. As you can see, this is basically the hardy chrome agar, the yellow one. And as you can see that there's like a big blob of purple color. All of this is basically the microbe that it is growing. It is growing in a garden culture throughout. So it assumed, because it was vulnificus, it assumed purple color. And the parahemolyticus, as I told you, it assumed a more teal color. These are the TCBS agars from which we took it. And then we plated it on the hardy chrome agar. The last experiment that we did was to test these microbes, all of them, all 50 of them, for a panel of 12 different antibiotics. All of these antibiotics are recommended by CDC for treatment of Vibrio and are commonly prescribed by the hospitals. So as you can see from these results, most of them were susceptible, and a small percentage of them, 11% of them, were resistant. And even smaller percent of them were somewhat intermediate. They were not exactly resistant, but they were not exactly susceptible as well. Now, this shows us that most of the Vibrio species are not exactly resistant to antibiotics, but some of them definitely are. This led us to our overall conclusion that out of Vibrio parahemolyticus and vulnificus, although they commonly inhabit the Chesapeake Bay, the vulnificus is present in really high populations. Most Vibrio species, although they were resistant to antibiotics, but small percentage of them did show resistance. Now, this is the start of antimicrobial resistant bacteria in the mainstream bay. If they continue to grow, this percentage can definitely drastically increase in the future. So they do pose a potential threat for creating superbugs in the Chesapeake Bay. So what we can do, or what is the future goal, is that we can definitely do an annual or quarterly or on a 6-monthly basis, a regular surveillance of these Vibrio species of the Chesapeake Bay to make sure that these resistant bacteria, or the superbugs, they do not increase. Okay, thank you for your time and patience, and here are my references.